students. So, today we are going to start something interesting where we are going to practice the objective with concept revision. Wherein Cell the Unit of Life we have dealt in detail also in the beginning of the bridge course sessions. So, today we are going to exercise the complete objective, all the objective from Cell the Unit of Life, wherein we are going to touch all the concepts. So, the first question is on plant cell. Wherein we have learnt plant cell includes 3 sets of organelles and the limiting boundary is cell wall. Plant cells limiting boundary is cell wall, and cell wall is connected, 2 cells are connected via plasmodesmatal connections. These are what? Plasmodesmatal connections which will help in connecting 2 cells, 2 plant cells. And what are the 3 modes of organelles we have learnt? Non-membrane bound organelles, single membrane bound organelles and double membrane bound organelles. Non-membrane bound organelle in the plant cell is only one organelle, those are ribosomes. And what type of ribosomes are present? We have both 70S and 80S type of ribosomes in a plant cell, right? And single membrane bound organelles include endoplasmic reticulum, 2 types of endoplasmic reticulum, rough endoplasmic reticulum and smooth endoplasmic reticulum, Golgi bodies and lysosomes, vacuoles, microbodies like peroxisomes, glyoxysomes and spirosomes are present. Got it? And double membrane bound organelles include chloroplast, mitochondria, and nucleus. Got it? So, a single plant cell will have 3 sets of organelles: non-membrane bound, single membrane bound and double membrane bound organelles. Non-membrane bound includes only ribosomes. If it is animal cell, we have ribosomes and centrioles, but here in a plant cell only ribosomes. As such, we have 70S ribosomes and 80S ribosomes. 70S ribosomes are present only in chloroplast and mitochondria, whereas 80S ribosomes you will see adjoined to the endoplasmic reticulum and freely present in the cytoplasm as well. And single membrane bound organelles include endoplasmic reticulum like RER and SER, Golgi bodies, lysosomes, vacuoles and microbodies. We have 3 types of microbodies like peroxisomes, glyoxysomes and spirosomes. Double membrane bound organelles include chloroplast, mitochondria and nucleus. This is a brief description on plant cell. Now you will be easily answering this diagram based question. Look at here. Diagram of a plant cell is given. Select the option with correct identification and one of the feature given with it. Okay. So, here if you can see B, which is marking what? RER, because you can see the extensions, the reticulum extensions from the nuclear envelope and it has ribosomes. Those extensions which have ribosomes from nuclear envelope, such organelle is what? Rough endoplasmic reticulum. So, he is saying what? B is smooth endoplasmic reticulum and concerned with the synthesis of starch, which is completely wrong, because first of all this is not SCR, this is RER, which is involved in what synthesis? Protein synthesis. If you consider this as SCR also, the smooth endoplasmic reticulum is involved in the synthesis of what? It is involved in the synthesis of lipids. Got it? Next, D is vacuole, which is covered by double layered tonoplast. Wrong. Here under smooth single membrane bound organelles we have learnt vacuole is surrounded by single membrane bound organelle. Vacuole which occupies 90% of the plant cell volume. It occupies how much? 90% of plant cell volume is covered by a single membrane and that single membrane is addressed as what? Tonoplast. The single membrane is tonoplast. Next, so this is wrong because he is saying it as double layered. Next, C, chloroplast which is having cristae in its inner membrane. Wrong. First of all, C is what? Chloroplast, that is correct only, but the inner membrane of the chloroplast is not cristae. Inner membrane of mitochondria is cristae. So, this statement is also wrong. Here in the question itself, he asked us to identify the correct statement regarding the diagram. And here if you see, A is plasmodesmata. This is concerned with the exchange of materials between the plant cells. Yes, A is plasmodesmatal connections, which is made up of the protein desmotubulin, and yes, this is involved in the connection of two plant cells. So, what is the right answer? Option 4 is the right answer. Clear with this question, ma? Clear, right? Yeah. And next, if you observe this question is from animal cell. Let us once quickly recall about animal cell also. Animal cell limiting boundary is what? Plasma membrane or cell membrane, and this also has three organelles, three types of organelles. One is non-membrane bound organelles, second single membrane bound organelles, third double membrane bound organelles. Non-membrane bound organelles of an animal cell includes ribosomes and centrioles. Ribosomes and centrioles are the two non-membrane bound organelles that you can come across. Single membrane bound organelles include all those organelles which are involved in the endomembrane system, like endoplasmic reticulum, both rough endoplasmic reticulum and smooth endoplasmic reticulum, Golgi bodies, lysosomes, and vacuoles are negligible here. Vacuoles are negligible, even if they are present, they are very limited. You can also consider vacuole, but you have to remember that vacuoles in an animal cell are limited. Next, double membrane bound organelles include the same chloroplast. Sorry, there is no chloroplast. Chloroplast is present only in the plant cell. It includes mitochondria and nucleus. You can see three major changes. The limiting boundary of a plant cell will be cell wall. Here it is cell membrane. Second change, centrioles are absent. Centrioles are amembranous organelles which are absent in a plant cell. They are present in the animal cell. Third point, vacuoles are negligible in the animal cell. They are present in the plant cell. Fourth point, chloroplast is present in a plant cell and it is absent in the animal cell. These are the three major points to be remembered. Now look at the diagram and question. Cell diagram of an animal cell is given. Select the option with correct identification along with the function or feature. We need to select the correct identification with the function or feature. If you see P, these are what? Golgi bodies. P is Golgi apparatus and this is mainly concerned with packaging of materials. Correct? Golgi bodies are concerned with what? Packaging of materials. And R, you can see two cylindrical rod like structures perpendicular to each other. Okay. R is centriole and is concerned with cell division in both plants and animals. No, in plants centrioles are absent. The function of centrioles in a plant cell, the function of centrioles is compensated by microtubular organizing complex. This is compensated by microtubular organizing complex. S is plasmodesmata. Yes, S is plasmodesmata and this is concerned with, S is not plasmodesmata because here you can see this is a nucleus and extensions of this you have what? Endoplasmic reticulum. Extensions of this endoplasmic reticulum are present. And these are what? Nuclear pores, not plasmodesmata. So, this will be wrong. And Q, yes, this is smooth endoplasmic reticulum and this is mainly concerned with the synthesis of proteins, he is saying. This is mainly concerned with what? Synthesis of lipids, not proteins. So, what is the right answer? Option 1 is the correct answer, right? Next question. Yeah, this is the question from the extensions of nuclear envelope, that is endoplasmic reticulum. A question from endoplasmic reticulum. Endoplasmic In the given diagram, nucleus is shown to be associated with cell organelles. Select the option with correct identification. We need to select this with correct identification and one of the features related with it. B, you can see, yes, this is rough endoplasmic reticulum. Steroidal hormone synthesis, wrong. B is RER, true, and here you can see the ribosomes which are adjoined to this. Yes, that is correct. But then RER is involved in the synthesis of what? Proteins, not steroidal synthesis. Next, D you see, D is Golgi bodies packaging of materials, wrong. What is this? Smooth endoplasmic reticulum, because it is in close proximity with RER and it does not have any ribosomes over it. How can you verify whether it is SER or Golgi bodies? Golgi bodies will have the cisternae like structure like this, cisternae like structure. Those are not the reticulum of structures, the tubular structures which are beside RER. These are cisternae like structures which are present next to the SER and RER. Next, A, if you observe A, which are these pores. This is nucleus and these pores are what? Nuclear pores. Nuclear pores. These are not plasmodesmata. A is not plasmodesmata. Transport of materials across the nuclear membrane. Function is correct, but this is not plasmodesmata. And C, if you see, C is what? Ribosomes. Exactly, C is what? Ribosomes, correct only. And this is involved in the synthesis of proteins. Ribosomes are purely involved in the synthesis of what? Proteins. So, option 4 is the right answer. This is all about endoplasmic reticulum. Once, let me give you a clear diagrammatic description. Nucleus has 4 components inside it. Nucleus has 4 components inside it. One is nucleoplasm, second nucleolus, third nuclear membrane, and fourth is nuclear pores. These are the 4 components. Got it? And here in this diagram you can clearly see about nuclear pores which are involved in the transport of materials, ions or molecules from the nucleus to cytoplasm or vice versa. Next, nuclear membrane. Nuclear membrane, you can see these are the extensions or reticulum of structure which is completely the extensions of nuclear envelope. Nucleolus is present inside the nucleoplasm which is involved in rRNA synthesis, and inside nucleoplasm genetic material is present, that is DNA. Clear, right? This is about the brief description of nucleus. Now, the extensions, reticulum extensions of nuclear envelope, if they are having ribosomes over it. One question from my end, how ribosomes are adjoined or connected to the endoplasmic reticulum? This is via protein called as riboforin, riboforin protein, where the larger subunit of the ribosome 60S, right, 60S is the larger subunit of 80S ribosome. 60S subunit is connected via riboforin protein, and these ribosomes are purely involved in the synthesis of what? Proteins. Next, close to this you have smooth endoplasmic reticulum which is involved in steroidal hormone synthesis. This is involved in lipid synthesis. This is also involved in detoxification of drugs. These are the functions of smooth endoplasmic reticulum. Clear with this, right? And then, yeah, about the structure of a chromosome. Structure of a chromosome. At the center you can see chromosome which consists of chromatids, and here how many chromatids are there? 2 chromatids, 1 and 2. These are the arms of chromatids, and you can see 2 equal arms of the chromatids, which is metacentric chromosome. Metacentric chromosome. And in which phase you can see the clear structure of chromosome? Clear structure of the chromosome is observed during metaphase. Clear structure of the chromosome is observed during metaphase. At the center there is primary constriction. At the center there is primary constriction. Adjoined to this primary constriction, there are 2 disc-shaped structures. These disc-shaped structures are known as kinetochores. So, a single primary constriction, that is centromere, will have how many kinetochores? 2 kinetochores adjoined to this. To this kinetochores only, spindle apparatus or spindle fibers are binded, and that will align, that will help in the movement of chromatids during anaphase, right. So, spindle fibers are attached to which region of a chromosome? Kinetochores. Next, there is secondary constriction above this part. The secondary constriction is known as satellite DNA, which has more of repetitive DNA sequences. So, in this chromosome they are asking us number of centromeres, number of kinetochores and number of arms. Which of the following table refers correctly to the chromosome? Number of centromeres, here how many centromere is present? Single centromere. So, there is 1 centromere is present, which is here. Number of kinetochores, a single centromere or primary constriction will have 2 disc shaped structures adjoined to this, these are kinetochores. Number of arms, how many arms are there? Single chromatid will have 2 arms, likewise we have 4 arms of the chromatids. So, what will be the right answer? Option B. Clear, right? So, you have 2 chromatids here, first chromatid and second chromatid. Each chromatid has 2 arms. Each chromatid has 2 arms. Likewise, total you have 4 arms in this chromosome. Got it? Yeah. Next question. Same type of question from chromosome structure itself. This is what? Primary constriction or centromere. This is secondary constriction. Above which you have repetitive sequences known as satellite DNA. What does A, B, C in the given figure of chromosome? What does it have? A is centromere, B is satellite DNA, C is what here? Secondary constriction. So, the right answer is option 3. Got it? Yeah. Next question. Match the following type of question based on the functions of the organelles. Match the column 1 and column 2 and select the correct option from the codes given below. Golgi bodies which are purely involved in packaging of proteins and lipids which are synthesized by endoplasmic reticulum. Packaging means what? Addition of sugar substances like sugar molecules, be it mannose, glucose. Adjoining to that you will be packing up, Golgi bodies will be packing up the proteins and lipids, and they are released out as vesicles. See, this is a nucleus, these are the extensions, here we have ribosomes. Let us suppose smooth endoplasmic reticulum is also present here. These are synthesizing naive proteins means inactive proteins, and lipids are being synthesized from smooth endoplasmic reticulum. These are taken from the cis phase of Golgi bodies, which is convex phase. Okay, this is cis phase, also known as convex phase of Golgi bodies. So here you have network of cisternae, inside which packaging of lipids and proteins takes place. Packaging, that is addition of sugars, wherein from the concave phase or trans phase, trans or mature phase or concave phase, what are being released? Vesicles are being released, and these vesicles are rich in hydrolytic enzymes. These are known as lysosomes. Few of the vesicles are transported to the intracellular targets and intercellular targets as well. Some of them will be moving to the plasma membrane, where in glycoprotein glycolipid composition is established. Those are synthesized from Golgi bodies itself. So this is the first process that happens in the endomembrane system. Here you see Golgi bodies, formation of what? Glycoproteins and glycolipids happens where? Inside the Golgi bodies. So yes, A goes with option 3. Next is lysosomes. Lysosomes help in digesting of or digestion of biomolecules. Lysosomes are rich in hydrolytic enzymes like hydrolases, carbohydrases, proteases, lipases. They are rich in hydrolytic enzymes and they are involved in digestion of biomolecules. Lysosomes, which is option 4. Got it? Vacuoles. Vacuoles trap the waste and excretory products. Vacuoles, which is the last organelle of the endomembrane system. The organelles which are involved in endomembrane system includes what? Endomembrane system includes all the single membrane bound organelles, which include endoplasmic reticulum, Golgi bodies, lysosomes and vacuoles. Got it? So till Golgi bodies it is clear to us. Lysosomes, we have different types of lysosomes, but as of now you remember that lysosomes rich in hydrolytic enzymes and they help in digestion of biomolecules. After digestion, all the cytoplasmic waste, excretory waste is deposited or trapped into the vacuoles. How they are sent to the vacuoles from cytoplasm? Via active transport. Via active transport. This is how all the functions of these 4 organelles are coordinated. As the functions of these organelles are coordinated, this set of organelles are placed in endomembrane system. So vacuoles, here you can see they trap waste and excretory products, which is option 2. Next, ribosomes, non-membrane bound organelles. This is purely involved in the synthesis of proteins. So what is the right answer over here? A3, B4, C2 and D1. Option 1 will be the right answer. Clear, right? Next. Here we go with next question, which includes match the following type and the membrane type. Okay. Mitochondria, which is double membrane bound organelle. Option 3. Because about different membranous organelles like amembranous, single membrane bound, double membrane bound, we have sorted out initially only when we are exercising about plant cell and animal cell. Mitochondria is a double membrane bound organelle. Lysosomes are single membrane bound organelles. Ribosomes, non-membrane bound or without membrane, that is option 1. And a nucleus, this is double membrane bound with pores. It has nuclear pores. We have also learned the components of the nucleus, right? So here this is option 4, D. So choose out the right answer. A3, B2, C1 and D4. Option 3 is the right answer. Doubts will be answered at the end of the session. If you are having any doubts, you can drop it in the comment box. Your doubts will be answered at the end of the session, not in the middle. Next question, this is on mesosomes. Mesosomes is a very important component from prokaryotic cell concept, where every year or every alternate year, either in NEET or MCET paper, you will be observing a question on mesosomes if it is from cell the unit of life. That important that concept is. So, here if you see, mesosomes are the infoldings of cell membrane, where mesosomes, what are mesosomes first? Where do you find that? We know bacterial cell has cell envelope. Can you see here? This is the cell envelope. Outermost, it has glycocalyx, followed by cell wall. Next, cell membrane. The extensions of cell membrane, the extensions can be tubular or in the form of vesicles or lamellar. Any type of extensions, be it tubular or in the form of vesicles or lamellar extensions. These extensions from the cell membrane towards the cytoplasm, membranous extensions, are addressed as mesosomes. These are known as what? Mesosomes. They help in cell wall formation, DNA replication and respiration, correct. They increase the surface area of plasma membrane, true. They are present both in prokaryotic and eukaryotic cells, wrong. They are present only in prokaryotic cell. So, 3 is wrong. Which of the following options are more appropriate? Mesosomal functions include what? Cell wall formation, DNA replication, respiration, daughter cell formation and DNA duplication, increase in surface area of plasma membrane and secretion as well. These are the 5 to 6 functions of mesosomes, which are very important. You will observe this in only prokaryotic cell. So, what is the right answer here? Option 1 and 2 are correct. So, option 1 is the right answer. Clear, right? Next question. Consider the following statements. Here the statements are again on mesosomes. Statements are again on mesosomes. Mesosomes are chromatophores. They are membranous structures in a prokaryotic cell. Chromatophores are pigment containing structures in cyanobacteria. That is true. Mesosomes, which are present in cyanobacteria, contains of pigments, and that is addressed as what? Chromatophores. Mesosomes and chromatophores are membranous structures in a prokaryotic cells, true. Both are observed in a prokaryotic cell only, because blue-green algae is a prokaryotic cell. It does not have a membrane bound nucleus. So, both the statements are correct, option 1 and 2. Option 4 is correct. Next. Yeah, this question you see, which of the following is found both in prokaryotic cell and eukaryotic cell? Semi-fluid matrix or cytoplasm, called cytoplasm, yes. The semi-fluid matrix of the cell which is observed both in prokaryotic and eukaryotic cell is cytoplasm, which is true. Next, selectively permeable plasma membrane is present both in prokaryotic and eukaryotic cell. 70S ribosomes are also present both prokaryotic and eukaryotic cell. In eukaryotic cell, chloroplast and mitochondria you can observe 70S ribosomes. Membrane bound nucleus is absent in prokaryotic cell. Genetic material DNA is present. If here they are specifying circular DNA or linear DNA, then we can observe the differentiation. But here he has mentioned genetic material DNA, so we cannot identify any differentiation. Chromatophores observed only in the prokaryotic cell, not in the eukaryotic cell. So, the right answer includes A, B, C and E. Option 1 is the right answer. Next question, last question of today's session, which is based on the discoveries of this chapter. asked in the year 2023. Endoplasmic reticulum, ribosomes and cell membrane, these three are given. Cell membrane proposed by, model is proposed by Singer and Nicholson, and that is fluid mosaic model. So, C, P and Y, these are matched. Next, ribosomes, protein synthesis and discovered by George Paliday. So, B, Q and X matched. Next, endoplasmic reticulum by Porter, involved in secretion. If you observe smooth endoplasmic reticulum, it is purely involved in secretion. So, A, R and W. A, R, W, B, Q, X and C, P, Y. Option A is the right answer. So, the key takeaways of today's session, apart from centrioles, cytoskeleton, double membrane organelles in detail, we have touched the major concepts from Cell the Unit of Life and exercised the objective. Like we have looked into mesosomes of a prokaryotic cell, and then we have looked into chromatophores. We have learnt in detail of a plant cell, animal cell. We have also exercised the questions on endomembrane system, and lastly organelles and their discoveries. Okay. So, with this today's session is done, and tomorrow we will practice few more questionnaire, few more set of questionnaire from cell cycle and cell division with concept revision. Mostly we will be exercising on meiosis 1. Yeah, thank you.